In the 1930s, people of America were engaging on the fun side of life. There was music and dancing and flappers, and Prohibition had just ended. The science world had just discovered something that would lead us to understand the brain and the cells in general in a whole new way.
Each spring, loligo squids migrate to the shallow waters off Cape Cod on the eastern coast of the United States. These squid provide neuroscientists at the Marine Biology Laboratory in Woods Hole, Massachusetts, with an opportunity to study the mechanisms of electrical signaling along the nerve axons. In the 1930s, scientists first started to take advantage of the loligo squid's giant axon for studying the electrophysiology of the nerve cell. Investigators can isolate an individual axon and insert an electrode into it to measure the axon's membrane potential and monitor its electrical activity. Loligo squids possess some of the largest nerve cell axons found in nature. The axons can reach 10 centimeters in length and are over 100 times the diameter of a mammalian axon, which is approximately the width of a pencil lead. The larger the diameter of an axon, the more rapidly signals can travel along its length. There are a variety of questions that researchers can address, including which ions are important for establishing the resting membrane potential and for initiating and propagating an action potential, and how changes in the membrane potential control ion permeability.
Once researchers could reliably generate and measure an actual potential, they could use the preparation to answer other questions about membrane excitability. For example, which ions are critical for an action potential? The three most plentiful ions, both inside and outside an axon, are sodium, potassium, and chloride. Do they have equal importance when it comes to the action potential? Because the squid axon is so large and robust, it was possible to extrude the cytoplasm from the axon like toothpaste from a tube. The emptied out axon could then be reinflated by pursuing it with pure solution of sodium, chlorine, and chloride. Thus, the ions inside the axon and in the bath solution could be varied independently. In this way, the researchers could show that the axon would generate a normal action potential if and only if the concentrations of sodium and potassium approximated the natural concentrations found inside the cell and outside the cell. Thus, they concluded that the cell components crucial to the action potential are the plasma membrane, sodium and potassium ions, and the energy provided by the concentration gradients of these ions across the membrane. All of the components, including the other sources of metabolic energy, were presumably removed from the perfusion.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and ATP provides the energy to change the shape of the channel. That in turn drives the ions through the channel. One phosphate group from ATP remains bound to the channel. The sodium ions are released on the other side of the membrane to the outside of the cell, and the new shape of the channel has a high affinity for the potassium ions, and 2 of these ions are now bound to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell.
These studies on the squid giant axon made an enormous contribution to our understanding of nerve cell excitability, and the researchers who made these discoveries in the 1940s and the 1950s, Alan Hodgkin, Andrew Huxley, received a Nobel Prize in 1963. However, it was years before the various ion channel proteins that they had hypothesized to exist would be biochemically identified. We now know the three-dimensional structure of many of these channel proteins, allowing us to marvel at the fundamental beauty of these molecular machines.